Hello, everyone. This is Victor Rush. I'm going to start the webinar. This is one of hopefully a series of webinars on integrating electrophysiology and fMRI. The first one is going to discuss hurdles and resolutions to integrating electrophysiology and fMRI. I am the Director of Technical Sales at TDT, and I've been here for close to 20 years. So I have a long and storied history with the company. For those who don't know, we build electrophysiology equipment. We have what's called a vertically integrated system for designing and manufacturing equipment for neuroscientists. So first thing is, when you're integrating electrophysiology and fMRI, you have really two very different types of recording systems. The first of these is basically picking up neural activity, electrical activity from the brain of an animal. So over here, we have a rat in a little box. It's awake, it's behaving, it's freely moving, and we can pick up neural activity, spiking activity. And we can also filter that and look at longer term activity, what we call local field potentials. On the right, we have fMRI. So the animal is head fixed and restrained in a tube. And we are going to be looking at longer term activity over the couple of seconds to minutes. And we are going to be looking at the whole brain as opposed to a very small section of the brain. So what I'd like to do in this webinar is provide you with an overview of fMRI and electrophysiology, a brief description of why I integrate the two, and I'm sure as neuroscientists you will be better at that than I am. The hurdles to concurrent in fMRI in electrophysiology and TDT solutions to that. So the real issue that we have is that we have several different types of recordings, and each one of them has different temporal and spatial and what I'll call field of views. So at the smallest level, we have single unit activity where the temporal resolution is very fast, on the order of a millisecond. The spatial resolution can be very small, depending on the type of electrode, and the field of view is very small. And as we expand out our electrical recording types, we go from ECOG to EEG, our temporal resolution becomes worse, but our spatial resolution and our field of view becomes better. And then we come into two new fields, which are the calcium imaging and fMRI. With calcium imaging, we have a medium-sized temporal resolution, sometimes excellent spatial resolution, in the order of tens of microns, single cells, and we can have a very large field of view. With fMRI, we have a larger temporal resolution, larger spatial resolution, and a greater field of view, again, the whole brain. And so these two things, fMRI and neural activity, provide us with a range of temporal, spatial, and field of view properties. This is an example of that. To the left is spiking activity. You can see that each one of these sections is 1 second. So what you can see to the left is very fast neural activity, firings of single neurons. In the middle is local field potentials across a broad number of channels and the neural activity there. And then to the right, the slower but larger area of the brain where we're looking at fMRI activity. So we know that there are these differences, and so why integrate the two? And again, you guys are better at this than I am, but one of the main things is to better understand the underlying architecture of the brain. Using fMRI and electrophysiology together gives you a better feel for what's happening. You also have the validation of the fMRI data, where you can determine the correlation between the fMRI signals and the local field potential or spiking data. And also You can look at the recording between the neural activity and fMRI activity from multiple sites and look at the spatial and temporal time course of signals. And finally, there's the validation of the BOLD data, which is understanding the relationship between the BOLD and the neural response, and also elucidating the underlying neural activity that drives the BOLD response. So what are the hurdles to the concurrent fMRI and electrophysiology? There are four basic ones: scanner artifact on the amplifier, contamination of the fMRI signal, visualizing the neural signals in real time, and then the real-time artifact rejection. So if we look at scanner artifact on the amplifier, the scanner artifact is huge. It can be on the order of hundreds of millivolts. And so you have an issue, which is that standard amplifiers to have a range of about 8 millivolts will saturate, and I'll show you what that looks like. And as you increase the input range of the amplifier from, say, 8 millivolts to 128 millivolts to the TDT's 500 millivolt, you also have an issue, which is trying to get a good reproducible range of signals from the largest to the smallest signal. And finally, you also have an issue with signal resolution. So with the TDT amplifier, you're going to see that we have a very large input range, a large dynamic range, and a very small resolution, or at least significant bit. Whereas with the other systems, you will find that you're losing signal quality or you have a problem with signal saturation. So you can see here, this is a 500 millivolt range. It's basically three times that of the 128 millivolt range, over 4 times that of the 64, and close to 50 times that of the 8 millivolt range. When you have a signal, like in fMRI, you have this very large scanner artifact, which is here, here, and here, over here. When it goes through a very small input range, like an 8 millivolt amplifier, the signal becomes saturated. So it clips on the top, and then you lose a lot of the signal as it recovers from that clipping. So over here, you have a 75 millisecond range. You will not pick up any signal after close to 100 milliseconds, where in theory, you should be able to resolve signals after about 45. Here's another example with a 7T scanner, a 6.4T scanner, and a 3T scanner. And you can see that depending on the amplitude of the artifact, you're going to lose more and more of your signal. So you have to have a large input range. Here's an example with a 64 millivolt input range, and you can see that even now I'm still saturating my signal and I'm not recovering completely. And if I go to actually the 3T, which has the largest scanner artifact, I still haven't recovered after several hundred milliseconds. When you go to 128 millivolt input range, which is here, you compare that to the 500 millivolt input range of the TDT amplifier, it still saturates in several cases. And so if you don't saturate, which is one of the things that the TDT amplifier will not do, you will get a better reconstruction of your signal. All right, so the next issue is really the dynamic range of the signal. So the difference between the largest and smallest signal. The worse your dynamic range, the harder it is to resolve these small signals. And this becomes important because if you're looking at things like local field potentials or even spike in activity, you may not be able to detect very small changes in your signal. So for example, here's a PZ5 versus a 64 millivolt amp. If you look over a 10 millisecond range, there isn't much difference. Same here. You can see small variations when you compare it to 128 millivolt range. But when you get to looking at the 1 millisecond time scale, you start to see that the signal is really distorted and smoothed out compared to that of the PZ5 amplifier. So signal resolution and dynamic range become very important as you increase your input range. And so what TDT has done is provided an amplifier that has a high input range, that does not saturate during scanning, and also has a large dynamic range with a DC amplifier to minimize signal distortion. The next issue is the contamination of the fMRI signal. If I have a fMRI It is not do well in response to materials that are magnetic or ferrous, and this is a good example. This is where a ferrous electrode was put into the brain, and you can see that there's significant amount of distortion. If you use a non-ferrous or fMRI-compatible head stage, you can see that there's less signal distortion. And so one of the things that TDT has provided is a non-ferrous head stage with a very long cable on it to minimize your distortion in the fMRI. And then the final issue is that you can get amplifier EMF artifact on the scanner. This is a 3T scanner, and you can see that there's a lot of activity here that should not be here. So you're going to lose your dynamic range of your artifact of your scanner signal. So to minimize that, what we have is an RF filter here that allows you to remove this, the artifact induced by the amplifier onto your scanner signal. And so here what we have is an example of how we've designed the systems to minimize any scanner artifact. So the first thing we do is we have our non-ferrous head stage and connected to an MRI-compatible electrode. And that goes via a very long cable up to 6 to 7 meters into the control room, and that's connected to our RF filter. The RF filter is tied to the ground on the scanner. All right, and this basically removes almost all of your scanner artifact, as you can see over here, and then they get processed by the RZ processor. One of the things that TDT provides you with, which is a real-time artifact rejection. And this is going to allow you to look at your neural signal while you're doing your recordings. And this is called the MRI Artifact Gizmo. This is a feature of our Synapse software. And what this allows you is it provides you with a series of tools for basically thresholding and recording around your artifact. So here we have a working window that you can change that's going to allow you to scale the area where you have your scanner artifact. You're going to be able to save the raw signal for doing offline analysis, and then you have the ability to save your signals for either single units, local field potentials, or both. And so here we have set our threshold. Here's my rejection window. And then these are the two different variable artifact removal windows that allow me to scale where my artifact is and then look at the signals around it. We also have it so that we process the single units and the local field potentials separately so that it minimizes what we call a ringing artifact from the filtering of your local field potentials. You can, of course, also bypass that signal, and that allows you to look at both the scanner artifact and also your signal. So here's an example of what happens when you try to record during your setup. You can see that you cannot really tell any of your local field potential signals while the artifact is on. Similarly, if you're doing spike sorting, you're going to see a similar effect where you're not going to be able to detect spike in activity during your recording session. And so here comes my artifact, and you can start to see that the signal characteristics become significantly distorted, and it makes it unable to do any analysis of your signal. On the other hand, if I use my MRI Artifact Rejection Gizmo, this is going to allow me to detect and remove that signal from the recording. It should come up in a second. Here, sorry. And you can see that you can see some of the signal. It's not great, but you can also detect some of the waveforms within that. You get a better response if you're looking at spike inactivity. So this is recordings. This is a rejected firing. And these are neurons that are being picked up. And right about now, we're going to start to see the artifact. You can see that the artifact rejects most of the spike inactivity, but I can still see neural activity within my recording. So I can actually look at firing patterns of the neuron during the scan. So those are the three things that TDT does and provides you with the ability to do integrated electrophysiology and fMRI. A couple of things, you have full access when you purchase a TDT system to our support. We have 5 people working in the support. Mark, who's got a master's in BME. He's the director of technical support. Miles, who has been here for over 3 years, an applications engineer, as is Chris Walters. The hardware all comes with a 5-year warranty and free remote desktop support. So where TDT ends, your research begins. And so I want to bring in 2 experts in doing concurrent fMRI and electrophysiology. One of them has worked on validation of denoising technique. This is Yuncong Ma from Penn State University. This is an example of how he has removed scanner artifact and the comparison between the electrophysiology after denoising in the scanner and what the signal looks like outside the scanner, and you can see it's pretty similar. So we have Jiayue Cherry and Yuncong Ma. Cherry is a senior PhD student in Xiangmin Liu's lab at the University of Michigan, and Yuncong Ma is a graduating PhD student in Dr. Nanyin Zhang's lab at Penn State University. So at this point, I'd like to pass the microphone over to them so that they can discuss the experience that they've had using the TDT system.
All right, there you go, Sherry. Yeah, we can see your slides.
Okay, sounds good. So I'm going to briefly introduce some part of my lab's work. So our lab is called Laboratory of Integrated Brain Imaging. So one of our focus is like we are interested in how the gut and the brain are interact with each other. Since there is multiple connection between the stomach and gut and brain, there are hormone. One of the most popular is about microbiome. Our interest is in the neuronal connection between the gut and the brain. And to aside such interest, we have to combine multiple techniques. We include functional brain fMRI, abdominal gastric MRI, neuromodulation include gastric electrical activity, vagus nerve stimulation, and we also include electrophysiology recording, like electrogastrogram recording, including the invasive and non-invasive EEG recording, and most importantly, the local potential and the single unit activity. So by combining all of these techniques together, we are able to answer the question of how the stomach and the brain interact with each other. So one of our focus is like assessing the stomach-brain interaction using the gastric electrical activity or the vagus nerve stimulation. So here is an example that we can electrically stimulate the stomach wall by implanting electrode on the stomach wall. And in the meantime, we can assess the neural activity in the brainstem nucleus or in the cortical regions. It gives us ability to acquire high quality neural activity. So in the meantime, we can also assess the functional brain fMRI and still with the stimulation on. So by using that, it gives us an opportunity to create a code simultaneous recording of the electrophysiology recording and the fMRI. So because the stimulation aligns the two signals together, by using that, we can see how the brainstem nucleus and how those responses affect the whole brain functional connectivity. So that's one example by assessing the brain response using the electrical activity, electro stimulation. We are also interested in the intrinsic stomach-brain interaction. So we have the stomach and the intestine, they are contracting and having their function continuously. And we also have the functional MRI to assessing the brain both activity in the brain. So we can use the electrophysiology recording to bridging these two signals. One bridging technique is the electrogastrogram. We can record the gastric electrical activity during the gastric MRI or during the functional brain MRI, and use the EGG as an intermediate signal, like to correlate and combine the gastric activity and the brain activity. Another bridging technique is that we can use the local field potential recording and the unit activity recording during the gastric MRI or during the functional brain fMRI. So by assessing the intrinsic stomach-brain interaction, we can reach out to real simultaneous recording of electrophysiology and the functional brain activity or the gastric activity. So here is an example that we can create an electro array to attach to the belly of the rat, and by using that, we can use the TDT system to record the cutaneous electrogastrogram, as shown here. And during this recording, we can acquire high quality gastric morphology imaging. So here is an example. This light half thing is the rat stomach, and by continuously acquiring the abdominal imaging with a faster sampling rate, we can see how the stomach contracts during the food digestion. And we can, by some image processing technique, we can extract the contraction waveform, and by comparing the simultaneously recorded EGG activity, we can see how the gastric electrical activity and the gastric mechanical activity coupling with each other. Since we get high quality EGG activity during the imaging process, we can also take advantage from the multi-channel activity recording. So by showing here is that we have the gastric electrical activity, which is the propagation electrical activity in the stomach. And by using the multi-channel technique, we can see how the propagation on the stomach showing on the cutaneous level. So here, when you see the arrow change during the signal, it's showing how the body surface potential changing during the propagation of the electrical activity on the stomach wall. So in the meantime, we can not only record the EGG activity during the gastric MRI to assess the mechanical changing of the stomach, we can also record the EGG during the functional fMRI to assess how the stomach activity interacts with the brain activity. So here is showing the simultaneous recorded EGG gastric electrical activity as shown in the red, and the bold activity showing the blue. We can see how these two signals coherent together, and using that technique, we can reveal high quality gastric synchronization map using functional MRI. So that's basically all our focus on the combining the electrophysiology recording and the MRI. And in the end, I would like to thank for all the audience that give us this opportunity to present our work and the books and give us this opportunity to discuss the challenge and opportunity in the simultaneous recording in the electrophysiology and the fMR. Thank you.
Thank you so much, Cherry. We will take questions at the end. Yuncong, I have your slide and I'm going to put it up now so you can take control of this if you have any questions. So I'm going to bring my screen back to sharing. Okay. Can everybody see my screen? Hopefully.
Yes, we're good.
Right.
So this is our recent work that showed in the Brain Initiative PI meeting. And you are free to register and download this poster to look at the details. So in our lab, we are interested in investigating the neural basis of resting state fMRI signal and the resting state functional connectivity. And in this study, we combine the electrophysiology and fMRI in sedated rats. In the experiment on the left, we put a 16-channel electrode array, 1 Michigan electrode in the anterior cingulate cortex to record the neural activity from the ventral to the dorsal part of this area. And the reason we choose ACC is to try to understand the coupling between the neural activity and the default mode network that involves the ACC area. And on the top left, we designed an animal holder that could perform this simultaneous acquisition experiment. And in the data processing in the middle, we designed a template-based method to regress out the MRI interference. So in that way, we could get both local field potential and multi-unit activity, which has been shown in this poster. And to validate this denoising method, we acquired data inside and outside the scanner under deep anesthesia, which induce burst suppression pattern. So in the validation figure that Victor showed before, we could you point to that? Can you see my cursor? Yeah, that place. Yeah, cool. So on the left is the data after removing out the MRI interference, which you could see periodic neural activity and intervals with no neural activity. And that shows consistent patterns comparing to what we acquired out of the scanner. So we could use this added validation for our methods. So in this way, we can get continuous FP recordings other than blanking out the MRI acquisition period. So in that way, we could get more information about this neural activation. And could you scroll down a little bit? Just to show some more validation results. Yeah, that's cool. So based on the literature, it shows that the gamma band power is tightly correlated to the fMRI signal. So we replicate the method, get the gamma band power from the ACC and convolve it with the hemodynamic response function and compute this correlation to the whole brain voxel-wise resting state fMRI signal. So on the right, we show the spatial consistent similarity between the two maps obtained from the gamma band correlation to resting state fMRI and seed map correlation. Okay. Oh, cool. And this result is consistent to previous literature report. So we are confident about this simultaneous recording platform. So we could move to our major project. So could you move to the result part? I think it's on the right top. So there's some figures here. Yes, scroll up a little bit. Yeah. Okay, cool, thank you. So on the right top, we get the ACC seed map, which shows the whole brain correlation to the ACC area. And also we, based on the traditional analysis, we separate the FP signal into 5 bands from the lowest delta to the highest gamma powers, and use them to compute the band-specific correlation to the whole brain resonant fMRI signal. And it shows that the gamma band is positively correlated to the resonant fMRI, and the other 4 bands show negative correlation, which also have been reported in previous literature. And because the TDT offers high resolution signal recording, we could further divide the FP signal into 1 Hz band powers, as shown in Figure C. So we could use those dedicated band definition to understand how this neural activity correlates to fMRI signal. And the result shows that the gamma band power from the 40 to 100 Hz, which we can go all the way to 300 Hz in FP, are positively correlated to the both signal. And the lower band under 30 Hz show negative correlation. And in Figure D and E, we are trying to go further because we found out some correlation between the different FP band powers. In that way, they may interfere and bias the traditional correlation analysis. So we introduce a generalized linear model to further understand the band-specific weighting on the fMRI signal. And in Figure F, we use some dynamic analysis called co-activation pattern that captures the spatial activation at the peak points of the fMRI signal. And in Figure E, that's fMRI cap patterns. And we could use those time points to align the FP signal and average those epochs to get the changes of the time course in different band powers that showed in Figure H. It shows that the gamma band increase and the other 4 bands decrease along the activation in the fMRI signal. And also we could get a full spectrogram aligned with the cap patterns in Figure I that shows those detailed neural activity. So in summary, that we combine this technique, the 2 modalities, to investigate the neural signature of the ACC network at the resting state. It shows the gamma band has a positive correlation to both signal, and the other 4 bands are negatively contributing to fMRI. That's part of our results as shown in the Brain Initiative meeting. And now we can get the multi-unit activity by blanking out the MRI interference. So we could get a full spectrum analysis that is part of our IS-MRIM talk by my colleague. If you want to look at it, you could search by her name and watch as a video. Thank you.
Thank you, Yuncong. Yes, and so actually, here are the 2 talks that they were describing. And they will be part of the multimodal fMRI session at ISMRM on August 13th. And so, hold on, they're up. Sorry. I was hoping that they would start from the right slide. So if you guys have any questions, feel free to ask them now. You can ask them to me or to any of our expert presenters.
All right.
Thank you very much for your attention.
All right, Victor, we do have one question from Thomas Marecci. He was wondering, Maurici, sorry about that. He was wondering, do you filter out artifacts from gradient switching?
So the answer is no. I mean, we basically, so if you want to do that, you're going to need to do that offline. Any artifact that is picked up is either going to be rejected, we're just going to throw it out and throw out any signal around it, or you're going to have the complete recording session and you're going to be able to do that offline.
All right. Let's see. I do not see any other questions. If anyone has any questions, feel free. Oh, electrodes.
Oh yeah.
There we go. Let me go open this up for a second. I don't know if you can see this or not, Victor.
Are you showing me electrodes? Or?
There we go. Here we go. Hi, could Cherry or Yuncong talk about their MRI setup? For instance, what type of screws do they use for the electrophysiology? What type of electrodes? And this is Livia de Mesquita Texeria. So let me switch over.
I think we should just have them all. Why don't we
just unmute everyone?
Yep.
Sorry about that.
No problem.
So we are using electrodes from Neural Nexus. They have MRI-compatible electrodes design. You could check their catalogs online or contact their sales support.
So the other thing is that there are a variety of labs out there that are making their own MRI compatible electrodes, and you have to sort of go out and ask them. We have a couple of groups that we might be able to pass that information on to any of the attendees. If they want to send us an email, viggyrush@tdt.com, we can reach out to them and get more information about MRI compatible electrodes.
I wasn't sure, Cherry, if you wanted to contribute anything else to that question.
Yeah, sure. Regarding the schools, yes, the current the common schools are using electrophysiology is not MR compatible. It will generate huge artifacts in the MR image. So we found a way to bypass that is by purchasing some plastic screws. But that plastic screws need to be manufactured a little bit because usually we use the screws for the reference and the grounding. Use as a reference ground electrode. So we need to manufacture a little bit to create some conductive component in the screws. So to let them not only secure the brain, and also generate some referencing and the grounding roles. So about the electrode, I think Neural Nexus is a great one, and our lab also have been working with the macro probes, and they also have great electrode which is MR compatible. So not only the electrode, but also the connectors, which right now the most commonly used connector is a magnetic connector. There is MR compatible version, so it works great. But for the electrode, we do test multiple material for the electrode. There is a platinum, and we also test what's that? Yeah, we compare multiple ones, and we found that the platinum and the IR, the TDT IR electrode works the best by our experience. And that's for all the commercial electrodes. If you would like to manufacture your own glass electrode, and I think that one works the best. There's no, even though a little bit artifact in the scanner, but you have to make your own, and it's fragile and easy to break. It's not suitable for chronic implantation, but if you are just interested in a acute experiment, I think that the glass electrode is the best. Yeah, that's basically all our experience in the electrode and the screws in the recording.
Okay, very good.
For the surgery part, so we use copper screws for securing the head implantation with dental cement to hold the electrode. And for the grounding wire, we choose pure silver made from WPI. It's very, very thin. You could use it to ground anywhere you want. It's quite easy, and we don't see big artifact. If you solder the wire, you will see big artifact because of the nickel.
Yeah.
Mm-hmm.
Yeah, and for the electrode, usually those commercially made ones have no artifact or impact on the EPI data. So if you want to localize the electrode, you could try to use very short echo time with longer TR for T2 weighted imaging.
Mm-hmm.
In that way, you could try to get the accurate position of the electrode in the brain. So that may improve your study.
Okay. Well, I do not see any other questions, unless anyone else has anything else to contribute. So I don't see anything else from the audience at the moment.
Okay. Well, I want to thank you all for staying on. And again, I want to reach out a great appreciation to Cherry and Yuncong for the detailed information that they could provide to this webinar. If you are all, if there are no more questions, we will conclude. And again, thank you for your attendance.